hour, if you would like that. Looks like you do. All right, so I'm just going to grab a blank sheet of paper and we'll just write away about. Alrighty. Switch gears here. Alright, here we go. Alright. I'll try and go. So, chapter 18. Alright, chapter 18 was, remember, about viruses and bacteria. So the first section is about viruses. So let's just review viruses here. Now there are two different kinds of viruses. The ones that infect bacteria only were called, and viruses in general, these guys look like this, remember? And they look like that. So what's the black part on the outside? What's that called? Capsid. And the inside is the blue, what would be? Yeah, it's, I'm going to put here, it's nucleic acid, because does it have to be DNA? No, there are DNA viruses and RNA. And they can be double-stranded, single-stranded DNA, double-stranded, single-stranded RNA. So that's one way that viruses are classified. All right, so that's the basic structure of it. And so these guys are unique in that they do one of two different cycles. All right, so this guy here, one of them, there's two things. There's the, what's the other one? Lysogenic. Which one of them kills the host cell? Lytic. Yeah, the lytic here. So this one kills host cell. Why is it important though? Why would the bacteriophage do this? What does it cut? As it kills the host cell, what has the host cell done for it first? Yeah, it made new viruses. Alright, so this one keeps the host cell alive. It has kind of a little different tactic. What does it do? It inserts its DNA into the bacteria's DNA. Into bacterial DNA. That's called a prophage. And then what does the bacteria do with it? DNA, it keeps it there, and so therefore the bacteria divides. And replicates then the viral DNA because it's part of its own DNA. So every time the bacteria divides, it now makes new bacteria with that viral DNA. So remember, bacteria divide very quickly. You can get millions within 24 hours. And so therefore, you can get lots of bacteria that are viruses, have the viral DNA. So this is this picture here. So this is the Lytic cycle where in this case here, the DNA is injected. And the DNA is used to make new viruses, and the host cell dies, and then all the viruses can go and infect new cells. This direction is the lysogenic cycle, where the DNA becomes part of the bacterial chromosome. All right, and so that's called the prophage. And so therefore, it divides and divides and divides, and makes lots of bacteria. At any given time, a particular bacteria that has the viral DNA, that viral DNA can pop out and it can enter the Lytic cycle. Alright, so that's the relationship there. Alright, so let's put here for underneath there that it can enter the Lytic cycle a virus, so that infects our cells, because these guys only infect bacteria. So these are a class called envelope viruses. And so these guys have the capsid and the genetic material in it. But in addition to that, it has an envelope, hence the name envelope virus. And the envelope viruses have these things called glycoproteins on the surface. So these are glycoproteins. This is an envelope. This is the envelope. This is the capsid. And this is your genetic material. Now, I'm going to draw over here, this is the host cell that it's going to infect. So, the glycoproteins in the envelope, what is it helpful for? To get inside the host cell. So, so there are proteins within the host cell that these glycoproteins bind to, and so then the whole envelope binds, and this is kind of like endocytosis, and inserts the capsid with the genetic material inside. So then what happens to the capsid? It gets destroyed, so here's your capsid proteins, exposing your genetic material. Let's say the genetic material is RNA. So then this RNA is used for one of two different things. To make copies. And so, here's all your RNA. Or the RNA can be used to make what? What is RNA used to make? Proteins, right? There are two kinds of proteins in an envelope virus. The first kind of protein are your capsid proteins. Alright? The second kind of protein are your glycoproteins. Glycoproteins, capsids are made on free ribosomes floating around in the cytoplasm. Glycoproteins are made on the rough ER. So I'm put RER, and here's your ribosome there. And the proteins, when the proteins are made, there's the chain of amino acids. They adhere to the inside, they bind to the inside of the rough ER. The reason why that's important is because, if you remember when we talked about the rough ER in proteins that are made of, they butt off in transport vesicles. Okay. And, yeah, they go to the Golgi apparatus, and then eventually will bind to the cell membrane. So I'm just going to draw that here. Here's the cell membrane. And so what happens when this occurs is that this binds with the cell membrane. So the cell membrane has these viral glycoproteins on the outside of it. All right? So that's the capsid, or the glycoproteins. Let's look at the capsid proteins come together and enclose around the RNA to make a new capsid for the virus. And what does this capsid do? It's going to come over here, and it's kind of hard to draw because I've already drawn this, but I'm going to attempt it. It's going to butt off through kind of like exocytosis, and this will have kind of like this, there's that here. And what you're going to end up with is a new virus that can now go infect a different cell. Alright, and so that's your new virus, and that's how that works. And that's how an envelope virus works. There's a special kind of envelope virus. HIV is an example. Those would be what? Retroviruses. So I'm going to put here retrovirus. These are envelope viruses. They are RNA viruses. That means that they have RNA as their genetic material. They have the enzyme, what did we just say? Reverse transcriptase. What does reverse transcriptase do? Takes that RNA, since the RNA is the genetic material, and uses it to make DNA, right? And then this is what's inserted into the host cell's DNA. So here, the RNA, so this is, retrovirus is a little bit different in that they're going to use that reverse transcriptase to make DNA, and then that DNA gets inserted. That's called a provirus. And HIV is an example. And that's basically viruses in a nutshell. Viruses get genetic variation from what? Mutations, all right? So if something mutates the genetic material, then you can get new strains of viruses. This is why we get different flu strains of viruses every year. All right. Let's see bacteria, 18.3 bacteria. All right, structure of bacteria. We just kind of went over this, but I'll just quickly do it here. It has the big circular chromosome. It has the plasmids. And then kind of goes with chapter 20, and I'm going to put in here, they also have restriction enzymes. Restriction enzymes are their natural way to destroy foreign DNA to kind of prevent kind of infections, I guess. And so we use that in chapter 20 with DNA technology. These guys replicate by binary fission. It's not mitosis, they don't have a spindle or anything, it's because they only have one chromosome. And this basically creates clones. You don't get genetic variation, bacteria do not get genetic variation by reproducing. How do they get variation? One way is the same way as viruses. Mutations. There are other ways. This is what has to do with our lab. Transformation. What does transformation mean? The bacteria does what?
Take up plasmids.
Takes up foreign DNA, all right? Takes up plasmids from other bacteria and so on, and so therefore can get new genes and new genetic combination. There's transduction. People can never remember this one. This is when a virus carries DNA from one bacteria to another. So it's when a virus infects one bacteria, and when the bacteria accidentally puts the bacteria's DNA in the virus, and then that new virus that has the bacterial DNA goes and infects another cell and injects that other bacteria's DNA into it. So that is transduction. And then the last form is called conjugation. This is when I drew this picture here. All right, this is called the sex pili. All right, and what happens is they can exchange DNA. All right, so this is one bacteria, this is another bacteria. They can exchange DNA. This is also called horizontal gene transfer. Because what's happening, normally we have vertical gene transfer, meaning when bacteria reproduce, you don't get any genetic variation. They get genetic variation from one organism to the next, swapping DNA. We get genetic variation by two parents having kids, and those kids get new combinations of genes. So it's vertical, meaning that the new combination comes with the next generation. This guy's here, new genetic variation doesn't come with the next generation. It comes with the current generation swapping genes and doing one of these things. Okay? And that is the basics of bacteria. I did already, for 18.4 is the operons. I had a video that I did last year about this. This is already on in the review folder in the playlist. So if you go in there, there's already a video all about the operons. All right? So we're running out of time, so what I will do next hour is make it chapter 20. Okay? Because this is already there. Okay? Sound good? You're welcome.
I just need to stop this.